The topic of this presentation is biotechnology and the ways that we can manipulate genes. This may seem like a completely different topic than our previous discussion of genetics and human inheritance, but in fact, biotechnology and genetic manipulation is one branch of molecular genetics. Molecular genetics is the study of the way genes are inherited on the molecular level, and in fact, how the DNA we inherit is normally used. The latter of those, how we use inherited DNA, is going to be the focus of this PowerPoint. The DNA we inherit is read by enzymes to produce RNA. Remember that DNA is double-stranded, and the two strands of DNA are held together by chemical bonds between the bases, the base being a portion of the nucleotides that make up DNA. 4 bases are found within DNA: A, C, T, and G, and they chemically bond with one another in the way that's shown in this image, A pairing with T and C pairing with G. When the DNA is read, the 2 strands of DNA are unwound from one another, exposing the bases on one of the 2 strands. I want to highlight that all of the DNA is not read, but rather segments of DNA are read one at a time. Those segments of DNA are the genes that we spoke about being inherited last time. The DNA is unwound and ribonucleotides are paired using an enzyme with the exposed DNA nucleotides on the unwound strand. A ribonucleotides pair with T DNA nucleotides, and C ribonucleotides pair with G DNA nucleotides to create the RNA product. U nucleotides pair with A DNA nucleotides when we are creating the mRNA transcript that is the end result of reading DNA. The mRNA transcript that is created is modified slightly in the nucleus before the mRNA moves into the cytoplasm. In the cytoplasm, mRNA is translated using a ribosome and helper tRNAs. In the cytoplasm, the mRNA is bound by a ribosome such that something called a start codon is revealed. The start codon is always a series of 3 nucleotides, A, U, and G in sequence. Once the A, U, G sequence is encountered, that sets the stage for translating the rest of the mRNA. It reveals then subsequent codons. A codon is a 3 nucleotide sequence on the mRNA that will be bound to a tRNA. Note that the highlighted AUG start codon is followed by a CCA subsequent codon. The mRNA assembles around this ribosome, and a tRNA carrying a methionine, MET, amino acid will bind to the start codon. Another tRNA carrying an amino acid, proline, will move into a little pocket on the ribosome such that an anticodon can recognize the CCA codon. Realize that the first G nucleotide in the anticodon will bind to the first C in the codon. The middle G nucleotide on the anticodon will bind to the C middle of the codon, and the U nucleotide in the anticodon will bind to the A in the last position of the codon. Once the proline amino acid is next to the methionine amino acid, the ribosome will form a chemical bond between those two amino acids, creating the start of an amino acid chain. And remember from our biological molecules topic at the start of the semester, a protein is a chain of chemically bonded amino acids. The code that is used to decipher the mRNA sequence is found on the right-hand side of this slide. The first nucleotide of the codon is found in the column on the left of the table. The second nucleotide of the codon is found across the top. And the third nucleotide of the codon is found in the column on the right of this table. A CCA codon would, using the table, produce a proline amino acid, exactly what is shown in the image. Knowing how we use DNA in our cells enables us to cheat the system and manipulate that DNA. That can be for excellent reasons. Creating genetically modified organisms is one of the manipulations that we can do. On the right-hand side of the slide, we see a gene, a sequence of DNA that is recognized by a specific enzyme. Recognition by this enzyme allows cutting of the DNA double strand such that a segment of the DNA, specifically a gene, can be removed. The manipulation part of all of this comes in when a replacement gene can be inserted where the original gene was removed. It's possible that the replacement gene can do something a little differently than the original gene that was removed. Once the replacement gene is in place, enzymes can repair the backbone of DNA such that the new replacement gene is incorporated into the original DNA double-stranded molecule, and we have a new sequence of DNA. The image of the fish on the right shows how genetic manipulation can produce a different organism. These are salmon, and the fish in the front is a normal salmon, an unmodified salmon, that normally only grows during certain times of the year. If we take out the genes, remove the genes that are responsible for regulating that salmon's growth, and replace it with genes that regulate growth in a non-seasonal manner, the fish continues to grow throughout the entire year. It creates a genetically modified salmon that sometimes has been referred to as a Frankenfish. These genetically modified salmon can grow larger in a shorter period of time because they grow year-round rather than only in the summertime, due to specific genes being replaced. Other food products can be manipulated in a similar manner. There are other applications of genetic manipulation. One of those is producing large amount of important proteins. The image on the left side of the slide shows us how we might do this. We take a gene out of a cell, and I will, for the sake of argument, say that this gene is the gene for human insulin. We remove this gene from a normal cell, and we insert it in a bacterial chromosome known as a plasmid. We cut the bacterial plasmid, and we can insert in the gene for human insulin. And using the same enzyme to glue the backbone back together, we can create a circular plasmid with an inserted human insulin gene. That plasmid is re-entered into a new bacterium, and those bacteria are allowed to grow to create a number of them. We can then allow those many bacteria to produce the insulin protein, and we can harvest that protein product, in this case human insulin, and use that pharmaceutically. Another application of genetic manipulation is the repair of DNA mutations that cause disease. This is called gene addition. We are not removing a gene and replacing it with a different gene, but rather we are adding a functional gene on top of a mutated gene. The way that this occurs in step one is that we obtain a copy of some functional DNA and we insert it into a virus. That DNA is incorporated into the virus's DNA. The second step, we remove the stem cells from the bone marrow of a patient that has some sort of DNA mutation. We allow the manipulated virus to infect that stem cell, and the inserted DNA that we put in that virus is incorporated into the DNA of the stem cell. That stem cell now has the added functional gene, and that stem cell then is returned to the patient with the DNA mutation. Now that stem cell grows and develops into other cells, terminal cells. The product of that gene, the inserted gene, is read and translated to produce the protein that the patient was unable to make with, in this case, her own DNA. This gene addition then can serve to produce proteins that might be missing as a result of these DNA mutations. A more recent development in DNA manipulation is the repair of DNA mutations by removing the mutated section and replacing it with a normal section. Perhaps some of you have heard of CRISPR technology. The diagram on the left shows you how CRISPR works. In this case, a patient, Susan, has a disease called sickle cell disease that results in an inability of her red blood cells to carry oxygen properly. We do know the DNA sequence of the gene that is mutated in Susan, and we can create a copy of that gene in the laboratory. The copy of that gene that's made in the laboratory is converted into RNA, and it is referred to as a guide RNA. It will guide the enzymes associated with CRISPR technology to the mutant gene. The guide RNA that was created in the lab is mixed with an enzyme known as Cas9. Sometimes Cas9 is referred to as molecular scissors because Cas9 is the enzyme that will cut out the mutated DNA. Cas9 and the guide RNA are returned to Susan or given to Susan, and the guide RNA directs the Cas9 enzyme to Susan's sickle cell gene. Cas9 cuts out the mutated section of Susan's gene, and her normal cell machinery replaces that missing piece of DNA with normal DNA. In this way, Susan's mutated DNA is repaired using CRISPR technology. This is referred to as gene editing, and this is simply another image showing you how that CRISPR technology is used to edit DNA. Forensics is another application of not necessarily DNA manipulation, but of our knowledge of how DNA works. Here we see two examples of something referred to as DNA fingerprinting. The way that this is used is we acquire some DNA from let's say, a crime scene. And we can cut that DNA into various fragments. The way that the DNA is cut produces a pattern of DNA fragments that is able to be visualized using some specialized equipment. In the image on the left, you see that the stripe labeled blood stain shows us the pattern of some DNA that was acquired from a crime scene. On either side of that blood stain stripe, we see samples of blood and DNA that was in that blood fragmented to produce a DNA fingerprint. These are potentially 7 different suspects that might have been responsible for the crime. Looking at the pattern of fragmentation of suspects 1 through 7 and comparing those with the pattern of fragments contained within the blood stain, which one of our suspects was responsible for the crime? The DNA fingerprint from suspect number 3 matches the pattern of the bloodstain found at the crime scene. Therefore, suspect number 3 is probably our culprit. Looking at the image on the right, we have 3 suspects, and we have DNA fragmentation patterns or DNA fingerprints of those 3 suspects that we can compare to the specimen that was found at the scene of the crime. Here, clearly the patterns between the fingerprint of suspect 1 and the specimen found at the crime scene match, so suspect number 1 is our perpetrator. Finally, the last application that I'd like to talk about is using DNA to individualize treatment for diseases. What's shown on the left is a gene pattern, and so on the right are lists of a number of different genes, and the gene pattern that is in normal tissue, those are shown in green, and the expression patterns of those genes in several samples from people with prostate cancer. If we look at just one of those patients' prostate cancers and we compare the gene expression patterns, the expression patterns of all of those different genes to the other prostate cancers, we can see some differences. I'm just going to highlight one. Here, our patient number, I believe 30, has a brighter red color indicating a stronger expression or overproduction of this particular gene. Overproduction of this particular gene can be pharmaceutically modified in this patient. So that might have an effect on this patient's prostate cancer. If we gave that same drug to one of the other prostate cancer patients, it wouldn't be expected to have as much of an effect because those patients' prostate cancers are not expressing this gene as highly. Therefore, we can individualize treatment based upon this patient's prostate cancer because this patient's prostate cancer is different from another patient's prostate cancer. This is the wave of the future in cancer treatments. These are just some of the ways in which manipulating genes can have an impact on our lives.